Hello and welcome to Jivology, a channel dedicated to making concepts in biology simple, easy to understand, and interesting. So, today we are going to look at different types of blood groups and blood transfusion in human beings. So, have you ever come across a situation where you were to donate blood and you wondered to whom will your blood go to, or if you were to receive blood, did you think of ever what if a wrong blood group is given to me? What is my blood type? Or do you even know that there are a lot of rare blood groups, and which are those? So, in this video, we are going to deal with all of these questions and a little more, where we are going to understand all about the blood groups in human beings. Before diving into blood groups, we need to understand the basic concept of antigen and antibodies. In this picture of the red blood cells, we can see certain round molecules which are attached on its surface. These could be proteins or carbohydrates or sugar. They are called antigen. My own antigen are safe for me, but when an antigen comes from outside as a foreign substance, it is usually harmful. It then induces an immune response and thereby it stimulates the production of antibodies. Now, what are antibodies? Antibodies are these Y-shaped cells that you can see. These float around in the plasma and they are produced generally to protect the body from the invasion of these antigen. So the antibodies would combine or bind with the antigen and then render them ineffective. Thus, an individual would always have antigens and antibodies in their blood, but the antibodies which it has would always be for those antigens which are absent in his blood. Easy to understand. Otherwise, if I have the same pair of antigen and antibodies, then they would combine and destroy my own blood. Now, why are we actually trying to study these antigen and antibody reactions? That is because our blood groups are classified based on the presence of specific antigen like the antigen A and B or even antigen Rh. And these antigen, as I said, trigger the immune reactions when there is a blood transfusion that happens. So that to ensure that the blood transfusion is safe, we need to understand these combinations of antigen and antibodies. We will look at them in much more details in the subsequent slides. How do the reaction happen? So an antigen and antibody reaction is called agglutination. So the antigens are the external foreign substances which come into the blood, and then they trigger the production of the antibody. Now generally we have genes which have the coding for specific antibodies, and then these antibodies are produced and they are found in the plasma. So the antibody has is generally Y shaped, and on top of these hands it has a specific um, you know a site which is called paratope, which is this site here, and this paratope uh, attaches itself to the epitope, which is the site of the antigen. Now this epitope of antigen and paratope of antibody, they are complementary to one another, just like a lock and key mechanism. So they fit into one another and then they form a bigger chain of molecules which is known as agglutination. So this reaction is known as agglutination, and once the agglutination happens in the blood, then there is a clotting and then there are serious problems that come into that happens to a person who receives these antigen antibody combinations. Let us see, based on this knowledge with us, let us see what are the different systems or different classifications of blood groups. We are familiar with a very common type, which is the ABO system, where we have heard people having A positive, B negative, and so on. So that type of blood group is called the ABO blood group type. That is one classification. This is based on the presence or absence of antigen A and antigen B on the surface of the RBCs. Now, depending on whether a person has just an antigen A or antigen B or both or none of them, there are four different types of blood groups: A, B, AB, and O. Now, I just told you that we've heard about A positive or B negative. How does this positive and negative come in? That comes from another system of blood groups, which is called the Rh blood type. So on the basis of presence of antigen Rh, there are two types of blood groups: Rh positive and Rh negative. So the ABO blood type was identified by Landsteiner, and Rh blood type was identified by Landsteiner and Wiener. There is also another system of blood groups, which is the MN blood group type. It is not a very common type, so we are not going to delve in details into this blood group type in this video, but just for you to know that the MN blood group type was also another type which was found by Landsteiner and Levine. Here, the human blood, when it is put into or transfused into a rabbit, there are certain antigens, antigen M and N, which produces certain serum antibodies M or N in the rabbits. If they are in the human being, there is no response. But when this blood, when the blood sample is given to a rabbit, that is when it triggers the release of serum antibodies M or N. Now, other than for academic purposes, this kind of transfusion of human blood would not happen with rabbits, and so you can understand why this MN blood group type is not a very commonly used or commonly referred to type, amongst humans. So as per the MN blood group type, based on the presence or absence of antigen M and N, there are three types of blood group: M, N, and MN. Now let us understand the most common classification of blood groups, which is the ABO blood groups. So ABO blood group is based on the presence or absence of antigen A or antigen B on the surface of the RBC. So depending on whether the antigen A is present or B, or both antigen A and B, or none of them, there are these four different blood groups. Let's look at each of them individually. So in blood group A, we can find that on the surface of the RBC, the antigen that is present is antigen A. Antigens are also known as agglutinogens. And in the case of a blood group A, the plasma would contain an antibody which is antibody B. Antibodies are also known as agglutinins. So we can see that the particular blood group has opposite types of antigen A and antigen B. Coming to B blood, you can see again that on the surface of the RBC there is antigen B, and in the plasma there is antibody A. So in case of the blood group B, the antibody A would combine with an antigen A in case of a wrong blood transfusion. Now what happens in group AB? The name AB suggests that it has both antigen A and antigen B on its surface of the RBC. So when there are both the antigens, what would be the antibody? Well, they cannot have an antibody, because if there is an antibody present in them, then that antibody would combine with its own antigen. So since they have both A and B antigen, they do not have an antibody. Coming to group O, though we call it O, basically it is zero, because there are no antigens attached at all to the RBC of the O type. So there are no antigens, and hence Their plasma contains both the types of antibodies, anti-A and anti-B. In the next section of the video, let us look at blood transfusion. This is a process where blood from one person is given to another. The person giving blood is called a donor, while the one receiving the blood is called the recipient. By now, you would have understood why it is important for us to know the blood groups of both the donor and the recipient and check whether they are compatible before doing the transfusion. But let us look at it through an example to understand it in much more details. So we have a recipient or a patient with blood group A, and we have two donors, one with blood group A and one with blood group B. Let us see what happens in the reactions to understand which donor would be more compatible for the recipient. In the case 1, the donor has blood group A. So he has antigen A on his red blood cells. Now, the recipient whose blood group is also A has antibodies which are of anti-B type. So, this antigen A and the antibody B cannot combine, and hence there are no agglutination reactions and the transfusion happens smoothly. But in the second case, which is the type B of the donor, here the blood contains antigen B, whereas the recipient blood contains antibody B or the anti-B agglutinins. So this type B antigen and type B antibody, they combine together to have an agglutination reaction, which leads to a situation called hemolysis, where as you can see that the blood flow is completely damaged. So how do we understand which blood group person can receive blood from whom? Because sometimes you can receive blood from more than one type of blood group. So if we look at the compatibility of the donor recipient table, we can see that a donor who has a blood group O can give blood to a recipient O, to a recipient A, to a recipient B, and to a recipient AB. And you know why so? That is because O does not have any antigens. Alright. Blood donor A has antigen A. So any blood group which has an antibody A cannot accept its blood. So O will not be able to accept A will be able to accept A, B will not be able to accept um, A, and AB will be able to accept A because A yeah, because A has antigen A in it, whereas here they do not have any antibodies. Similarly, when it comes to B, O will not be able to accept B, A will not be able to accept B, but B and AB will be able to accept the blood from the donor whose blood group is B. Moving on to AB, O will not be able to accept AB, A will not be able to accept AB, B will not be able to accept AB, only AB will be able to accept AB blood group. So if we look at this table, we can understand that the blood group O, a person who has an O blood group will be able to give blood to almost any other blood group type, and a person who has AB can receive blood from any other blood group type. So let's move on to the next type, which is the RH blood group. So the RH blood group is where we get the positive and the negative. So there is an antigen called RH, which is again present on the surface of RBC. Now, depending on whether the antigen RH is present or absent, the blood groups can be RH positive or RH negative. Now, RH comes from the word rhesus. Rhesus monkeys are the ones where um, there was an experiment which was done where the blood of the monkey was used for immunizing the rabbits. So the resultant antibodies in the rabbit's blood that agglutinized the monkey's blood, and the same blood from the rabbit also did the same to the humans, which means that the humans also have the same factor which the rhesus monkeys have. And this factor was known as the rhesus factor. And those humans who have the rhesus factors are known as Rh positive. So to avoid agglutination, we must make sure that apart from checking the ABO compatibility, we also need to check the Rh compatibility, that is whether the person is Rh negative or Rh positive, because Rh negative person should be given only Rh negative blood. An Rh negative person will not be able to uh, take Rh positive blood, because Rh negative person will have an antibody towards Rh positive. Let's look at the compatibility for the donor and recipient table, where we are looking at a combination of both ABO and Rh. So we have seen that O blood group can be given to all the different types. But when it comes to O positive can be given only to O positive, A positive, B positive, and AB positive. It cannot be given to O negative, A negative, B negative, and AB negative, because all these negative blood groups have an antibody called anti-Rh positive. So which means that if this blood comes in here, there will be an agglutination reaction. Similarly, A positive blood can be given to A positive and AB positive, and to nobody else. B positive can be given to B positive and AB positive can be given only to AB positive. Whereas when we look at O negative blood does not have any Rh factor in it, and hence it is safe to be given to anybody. So O negative can be given to all of these 8 different types of blood groups. A negative can be given to A positive, AB positive, A negative, and AB negative. Likewise for B negative as well. And when it comes to AB negative, it can be given to both AB positive and AB negative. So now this brings us to the concept of what is called a universal donor and a universal recipient. We can very clearly see that O negative is the type of blood group which can be given to anybody else. So O negative blood group can be used for transfusion for any other blood type. It is safe to use O negative for anybody else, and hence O negative is called as the universal donor, uh, which is also why O negative blood is used for emergency transfusions and also for small babies where the immune system is still not fully developed. Then there is universal recipient, which is AB plus or AB positive. You can see AB positive can receive blood from all the other different types of blood groups. So AB positive is known as the universal recipient. We also spoke about rare blood groups. What are rare blood groups? So we have now seen that there are antigen A, antigen B, and antigen RH. But apart from this, also there are more than 600 different types of antigens which are available in our human blood. If there is a person who does not have one or a combination of such antigens, in that case, such a person's blood is considered to be rare. So which means that a rare blood group is one which lacks either a single antigen or a combination of antigens which are otherwise generally found in majority of the population. So what would happen if they do not have this kind of an antigen? Which means they will have an antibody against it, which means that they will find it very difficult to receive blood from any other blood group, because whichever blood they receive would have this antigen, and they will have an antibody to go and do an agglutination reaction to that blood which they are receiving. Now, if in case you happen to have a rare blood group, what should you do? One, let your siblings and other relatives know about the rare blood group that you have, because it could so happen that they also have the similar blood group, because blood group is inherited through genes. The blood groups are decided through genes, so the inheritance pattern of blood group is genetic. So if you know that there is another relative, then you have a fallback option in case there is a need for a blood transfusion at any point in time. Apart from that, the blood banks generally have rare donor programs where they identify and maintain contact details of these rare blood group individuals. So you could get yourself registered there so that you become a part of the community, and it becomes easy to access your blood group or your unique blood group, which in case of an emergency. Now let's see a few different uncommon blood group types. Um, One is a Bombay blood group and the other is golden blood. Now these are two very, very rare types of blood groups. In case of Bombay blood group, uh, now we have seen that there is an antigen A, antigen B, which is present in most of us or absent amongst people. But apart from that, there is also a major antigen in the body which is called the antigen H. So all ABO blood group individuals have an antigen H available in them. But people who have Bombay blood group, they do not have antigen H. Hence, they have an antibody H. And because of the presence of this antibody H, they cannot receive the blood from any of the other blood groups A, B, AB, or O. If by chance such a blood transfusion happens, then it causes an acute hemolytic transfusion reaction, and it can lead to serious complications. So this Bombay blood group is present in 1 amongst 10,000 people in India. So we need to be aware of our blood group very well, because the absence of antigen H, uh, when we go further and study the biochemistry of the antigens um, present in the blood group, we will know more that antigen H is required for having antigen A and B attached. So on the antigen H, there is a chemical molecule which gets attached, and because of which an antigen A or an antigen B is formed. So if antigen H is not available, then antigen A and B will also not be available. So phenotypically, or through an external appearance, this person may be thinking that he is O positive or O negative. So they could appear as O because A and B both are absent. However, on further deep delving, we know that the person does not have even H in them, and hence the person belongs to Bombay blood group. Now then we have another thing which is called the golden blood. Golden blood is Rh null blood type, which means that this person does not have any of the Rh antigens. Now you may say that absence of Rh antigen is Rh negative, but here there is a slight difference. An Rh negative person does not have Rhd antigen. He has all the rest of the Rh antigens. So Rh antigens is a group of antigens of which Rhd is the one which is absent, and that makes a person Rh negative, but he would otherwise have the remaining Rh antigens in him. But a person with golden blood does not have Rh at all, so he would be Rh null. This is the rarest blood group in the world. It is believed that there are less than 50 people in this world who have this blood group, and of which while I was researching to make this video, I found that many places there is a mention that there are just about 9 people in this world who are active donors for this blood group. I do not know how much true or false that is, and hence I preferred not to use it in my literature here, because the data was from a few years ago. The number might have changed now, but basically what we're trying to say here is that for the golden blood, there are very, very few people in this planet who have this blood, and of which very few are active donors. This kind of a blood group was found in Aboriginal Australians for the first time, and since it is very, very difficult to obtain donors for this blood group, this makes the blood group very, very valuable, and hence it is known as golden blood. So this brings us to the end of our video. Let us summarize what have we seen so far. We went through the basic concepts of antigen and antibody, where we saw that the antigen are foreign substances which are generally harmful, and they trigger an immune response, because of which an antibody is produced, which helps the body to protect itself from the invasion of the antigen. Then we also saw the different systems of blood groups. The most common one is the ABO system and the RH system. And then we also saw about the MN type of blood group. Within the ABO type, it is the presence or the absence of antigen A and antigen B which determines the blood group. If antigen A is present, it is called A group. B is present, it is called B group. If both are present, then it's called AB group. And if none of them are present, then it is called the O group. The RH type of blood grouping is decided based on the presence or absence of the RH or the rhesus factor. If RH is present, the person is considered as positive. The RH absence, the person is considered as negative. So a person with the A group and an RH positive would become an A positive. Talking about blood transfusion, here we looked at the different compatibilities between the groups, and we came to understand the universal donor, which is the O negative, and the universal recipient, that is the AB positive. The O negative blood group can be given to all other blood groups, whereas A positive blood group is the one which can receive blood from all other different types of blood groups. We also saw the rare blood groups, where a rare group is one where a person lacks one or a combination of antigens which are otherwise generally present in the general population. We also saw two very, very uncommon blood groups. One is the Bombay blood group, where a person does not have antigen H, and then we also saw about the golden blood, which is the rarest type of blood group on earth, where a person does not have the RH type. It is different from RH negative because here the person does not have any of the RH antigen, whereas in RH negative the person does not have only RHD. So I hope I have been able to give you some information regarding the blood groups and the blood transfusion, and it has been helpful to you so far. If you do have any questions, please feel free to write or comment in the comment section, and I shall revert to you. Thank you so much, and see you in my next video, where I'm going to talk about the inheritance of blood. And if you like this video, please do like, share, and subscribe my channel. Thank you so much. See you again.